of the presentation, just to go quickly through some very general basic concepts. So yesterday we were talking about this, how this DNA code, the sequence of nucleotides, it's could be similar to a keyboard in a piano, where every key has a specific sound, or every nucleotide has a specific information. If we change that, we may end up changing completely the sound in the piano and the message in the DNA. So I'm gonna go here just to remind you that something very important is that during embryonic development, a single cell undergoes a lot of divisions so that we can have a complete organism form. And those divisions are, during each division, the DNA has to be replicated, and that large amount of replication that occur in DNA are the increases a lot the probability of getting mutations during the process, and we're going to talk about that today. So I was telling you yesterday that these mutations of the DNA is something that occurs normally, naturally. There are mutations, and that they give rise to this diversity in life that we have. So it's something that it's good for us that the DNA can mutate. It's good, but then can also be not very good for us if it happens under another circumstances. So if we think about the gene, so we have the chromosomes. If we go deep into the chromosome, we can see how it's organized in there are genes along the chromosome arms. If we go further into 1% of the chromosome, we can find like 4, 5, or 6 genes depending on the chromosome. And then each gene is also can also be divided into subregions that are, in this particular case, we see the regulatory DNA sequences, exons, introns. So the DNA at the end, it's just a series of nucleotide pairs that are organized in a such manner that it contains a code. So as you can imagine, any changes in that code will change the function of the cells of that particular gene. And basically what we're going to talk about today is mutations. So mutations are changes in the DNA-based sequence. And these changes in the DNA that happen in the DNA can be classified in different various ways or different manners. So here we have few ways of how these mutations can be classified. First, we can classify them as point mutations when it involves only one single nucleotide pair, deletions or insertions when it involves, we're going to see a little bit an example later. They can be spontaneous or induced mutations. They can be somatic or germline mutations, and it can be also classified as autosomal or X-linked mutations. So point mutations, insertions, and deletions occur when, yesterday we were looking at this, sorry, Monday we were looking at this picture where this particular position here, the GC pair, is changed either by an AT pair here, where this is a point mutation because there's only substitution of one position of the DNA sequence. And these point mutations can be classified either as transitions or transversions, depending on the type of nucleotide that is changed. And then we have the mutations that involve more than one pair. Those are called deletions or insertions. So deletions, basically, there is a part, a fragment of the DNA, a certain number of nucleotide pairs that are removed. It can actually be only one pair. Often are more than one base pair that are removed. And insertions where new nucleotide pairs are inserted into that original sequence. Then when we think about spontaneous or induced mutations, so I think on Monday we were also talking about, so what are spontaneous mutations and how, or what can induce the appearance of these mutations? And on Monday, you were telling that maybe DNA replication mistakes, when the DNA polymerase can actually make mistakes, and then that will end up in introducing a wrong base pair that will finally be established as a new base pair in the DNA. That's a mutation. We can also have defects in DNA repair mechanisms, and then we will see a few more examples of that. But when we talk about induced mutations, what do you guys know about induced mutations? How can induced mutations? What do we mean when we say induced mutations? Anyone has an idea?
Is that a general way how to study like a phenotypic response to see what gene actually codes for something? Like scientists could do that in a lab.
Right. So do you think there's any induced mutations that occur in nature? I mean, mutations that are induced in ourselves by something that is around in nature that we are exposed to?
Mutations can be induced by like UV light.
Exactly. So basically, there are radiations that we are exposed to, like UV light, and sometimes we can be exposed to other types of radiations. And also, we have other types of um, factors in the environment that are like chemicals. As you know, there are many chemicals that are now known, and they are actually known as mutagens, because if we are exposed to those or to large amounts of those chemicals, they can actually alter the DNA structure, and then that will result in mutations. And there are actually also infectious agents that can induce mutations in our DNA. So as you can imagine, we are exposed every day to a lot of factors that can induce mutations in our cells. So fortunately, our cells have systems to repair and to repair those errors or those induced mutations. Um, so the spontaneous mutations occur at very low rates in organisms and different organisms. Here's an example of a few organisms where if you see the estimated mutation rates in bacteria, it's one mutation every 100 million divisions. But if we go to mice, the estimation is that there's one mutation every 10,000 divisions. And in Drosophila and human, it goes from 1 in a million to 1 in 100,000 divisions. So it means that every 100,000 divisions, there is a probability that one mutation will be established. So that's a relatively low rate, but it can be increased by the presence of these factors that I mean, spontaneous mutations can, in combination with mutations induced by external factors, then can give rise to more frequent mutations. And as you can imagine, if the more exposure we have to these factors, the highest probability to getting these induced mutations. And yesterday we were talking about these experiments where there was this hypothesis that whether the bacterial resistance to antibiotics would be like a physiological response. It means when you expose bacteria to the antibiotic, they will, in response to that, they will generate, let's say, mutations. And the other hypothesis is that this resistance arises from random mutations. It means in the population of bacteria, there will be random mutations appearing here and there, and then some of those mutations will give rise to resistance to the antibiotic. So the experiments basically were they cultured bacteria in different plates or tubes, and they exposed them to antibiotics. And if the first hypothesis was true, that the bacteria will generate resistance because of the presence of the antibiotic, one will expect to see more or less the same rate of mutations appearing. It means in every plate that we have here, we should see more or less the same number of resistant colonies. And if the other hypothesis is true, then some of the bacteria will have mutations that will render them resistant, and then we will see different number of colonies. That is, it was what they observed was this. In each different plate, we have different numbers of colonies that were resistant to the antibiotic. That the conclusion of that experiment was that the mutations are randomly appearing and not pre-existing or not in response to the biological response. And another nice experiment, this one where they also have some bacteria growing here, and among those colonies, there was one that is resistant. You take it like a stamp and you stamp it in another plate, and you put those that plate in presence of the antibiotic. Then you see that the resistant colony, the resistant bacteria, will grow. And if you repeat that many times, you can see how the bacteria or the colonies are exactly in the same position, are replicated in every plate. So it means that the original bacteria or the original colony was resistant, and if you stamp it in a different plate, that particular colony will be replicated in all these plates. So that means that the mutation that giving rise to the resistance was already present in the original culture. And then we were saying on Monday also that these spontaneous mutations are due to defects either in the DNA replication mechanism for the DNA polymerase adding a wrong nucleotide, or defects in the DNA repair mechanisms. And then something interesting is that when the DNA replication goes much faster than the DNA repair, then we increase the probability of getting mutations, because then the cell doesn't have time to repair all those mistakes that the DNA polymerase is inserting. And as an example, I was telling you on Monday that when we look at the embryonic development, and particularly to the germ cell differentiation, during the first step, we get these primitive strict cells that are the ones that are going to be give rise to the germ cells. So here, during this stage, the cells are dividing a lot, and they may get some spontaneous mutations. Then we go to the second stage, where there is this differentiation in male and female progenitors, and they will also go to several rounds of division, and they will they have the probability to get in some mutations. But then, when they reach this stage here, in female, the number of oocytes is already established, and they no longer divide during after birth. So there is no longer division of oocytes after birth. Whereas in male, these spermatogonial stem cells are renewing all the time, and then when the sexual maturity is reached, they will start proliferating at a very high rate. So all these sperms have a much higher probability to getting mutations than oocytes. So, and there are very nice studies now using a single cell sequencing where they did actually demonstrate that sperms have a lot more mutations than oocytes. And this is because they just divided more times. So there are some natural processes occurring in the cell that also may contribute to these DNA mutations. And we're talking about these two chemical reactions, that is depurination and deamination. So basically here we see how these two rings are removed, and there's an empty site here, or deamination, this amino group here is lost. In any case, what happens is that when the replication occurs, so this nucleotide is no longer going to be recognized as a guanine, because now, or cytosine, because it has changed its chemical nature. So in this example here, the deamination example, we have a CG pair here. So the cytosine here, it's going to be deaminated, and it will result in a uracil. And during replication, so there will be one copy carrying still the uracil and one copy that will be similar to the original one. But in the next round of replication, we will get this mutant sequence here, because now this adenine here, it's going to be paired with the thymine. And so we have this mutant sequence here, and this sequence that is not, it carries still the uracil. So this is a way how mutations are established, just a single change in the structure of the molecule here. Now it's going to be recognized as a different one, and then in the further rounds of division, that mutation is going to be established. Similarly, we have mutations that are induced by like X-ray exposure or UV light exposure, and X-rays can break the DNA, and then DNA can re-ligate by losing this fragment. And then we have, as a result, we have a deletion here. We have UV light, which produces the covalent binding between two thymine dimers that are next to each other. And that structure, of course, can be recognized as an abnormal structure in the DNA. And we also have the oxidation that can occur, like in this example, in the one in where this oxygen group is added here. And then again, as in the previous example, when the DNA replication occurs, that oxidized guanine is going to be recognized as not as a guanine, but it's going to be paired with an adenine, as you can see here. And again, in the second round of replication, we're going to get an established mutant sequence here. So all these things happen naturally well, not naturally, but the first deamination and depurination, it happened in our cells all the time. And if we are exposed ourselves to UV light or to X-ray, we can also get these abnormalities. And oxidation is also happening in our cells. So as you can imagine, if we put all this together, there are many factors, endogenous or not, or environmental factors that can induce mutations in our cells. So this, there's another, I think Tere was talking about that, this on Monday. So we have these tautomers that are bases that are a little bit different from the original ones, like this immunoform cytosine here, where the structure of the cytosine changes a little bit, and then the way it forms the base pairs, the hydrogen bonds are different, so that can give rise to the pairing of abnormal nucleotides. And so something important is that, and this is again the same. So just because of these small changes in the structure, the nucleotide is recognized as a different one, and then we are going to having again we're going to have this mutant sequence. And there's another a bit more complex probably type of mutation, because it involves more than one nucleotide base pair, sorry. And this is there's in our DNA there are unstable trinucleotide repeats. And that means that you can have CGG, this trinucleotide, repeats many times in a DNA sequence. And what we know now is that these nucleotides are unstable, and we're going to see why they are unstable. And also they are the length of those repeats, it's related with many diseases. So here you have an example with the wild type alleles where we have less than 56 repeats of CGG. Then we have what we call the pre-mutation allele, where instead of less than 56, we can have between 56 up to 200 copies of that trinucleotide. And then we have the disease-causing alleles, where we have more than 200 repeats of these CGG repeat. So that's very, as you can imagine, if you have few copies of a trinucleotide, and if that's going to be translated into protein, you will have 5, 6, or 7 amino acids. Then if you extend that to 200, you will have a lot more amino acids in that region of the protein. And as an example, we have the Huntington disease, which is caused by this increase in the number of CAG trinucleotide, as you can see here. So at the DNA level, there are 6 to 28 repeats of CAG. It's what's considered normal. Then we can have the pre-mutation stage where we can have up to 35. And then if there are more than 36 and up to 120 repeats of CAG, that's the mutant, and that's related with the disease. And actually what happens is that this CAG sequence encodes for this glutamine. And then if the protein that is synthesized by this sequence, when there are 36 to 120 repeats, will have a lot of glutamines. That's toxic for the cell. This is just one example how this increased number of repeats can be related with the disease. And so how this increase in the number of repeats occurs. So it's something that is called like the slipped replication, let's say. So basically, because of these sequences are repeated many times, they can form these kind of loops where they basically, because they are homologous, they can actually form like bind to each other, like form structures that are not read by the DNA polymerase. So the DNA polymerase will slip that region, and then we add more nucleotides here, and then when that strand will be put back again into double-stranded, you will have an increase in the number of trinucleotide repeats. So as you can see here, you can go to an increased number over the rounds and rounds of replication. You will have every time more and more copies of that trinucleotide. So then there are these induced mutations that are, we were saying, we were already see some how DNA can be altered by UV light or by X-ray exposure. And also we have these mutations that are chemical reagents or chemical substances that can actually bind to the DNA. And that binding to DNA will alter the structure of the DNA. And if they are present during replication stages, that can interfere with the DNA replication, and then we can it can give rise to abnormal replication and to the introduction to different nucleotides and therefore mutations. So I'm not going to go into detail on that, but just so DNA damage can be reversed, and also the damaged bases can be removed and replaced by basically two mechanisms that are the base excision repair and the nucleotide excision repair. So here, as an example, we have, as you can see here, this red nucleotide has been deaminated, and then that will be released. That fragment of this strand will be removed, and then it's going to be, again, the DNA polymerase will fill it up again, fill that gap. So basically what happens is you remove the uracil, that is the one that was deaminated. There are some, the DNA will be cut at the backbone, and then some exonucleases will remove a little bit larger part of the DNA, and then the DNA polymerase will come and will fill it up again. And finally, a ligase will fill up this little gap here. So this is what's called the base excision repair. And then we have the nucleotide excision repair. In this example here, we have UV light inducing thymine dimers. As you remember, I mentioned you that these thymine dimers are formed by the linkage of the two thymines that are close to each other. And of course, there's a deformation of the structure of DNA. Then there are some endonucleases that can come here and also remove part of the DNA. And then once again, the DNA polymerase will come and fill the gap So in any case, basically we need enzymes that can recognize the DNA at the position where it's been damaged. There will be these endonucleases that will cut the DNA, and then the DNA polymerase can come back and fill the gap, and the ligase will finally ligate the new strand with the previous strand. So these are two mechanisms that are very important for repairing DNA, abnormal DNA structures. And we also have these recombination processes that can occur in our DNA. So here, just as an example, very quickly, that we can have a gene where we have a mutation here, M1, and we have a mutation here, it's M2, in the same region of the same gene, and then those two strands can recombine, and we can end up with a one strand that has both mutations and one that has no mutations, like the wild type. So this is a very important process also that may help to remove or to establish the establishment of DNA mutations. So if you think about the gene, so now the gene is like a linear set of nucleotide pairs that is divided into subregions, like the promoter region, coding region, non-coding regions that are important for the regulation of the transcription. Genes can also be organized as a monocistronic genes, where only one promoter, one gene, or polycistronic genes, where we can have one promoter region and several coding genes for different proteins in the same region. So as it happens in bacteria, for example, maybe you heard about the operons in bacteria, where you have one regulatory region and you have 3 genes in tandem. So when the transcription occurs, all of those genes are going to be transcribed, and the proteins coded for those genes going to be produced at the same time. And in eukaryotes, we have a bit more complex genes, where we have, again, the promoter region. The transcribe region can be divided into intronic and exonic regions, and those regions, when they are transcribed to RNA, the RNA can further go splicing, so that some introns are included and some not. So in this particular, I just want to point it out that there are like 2 major regions that I'm going to ask you to keep in mind. So the regulatory region, where we have the promoter, silencers, and enhancer, and the coding region. And for me, let's think about the promoter region as this is the region of the gene where the information of that region defines when is that gene it's going to be expressed, where, when, and how many copies of that gene at a certain time are going to be produced. So all the information to define those processes it's in the promoter region. And so if we think about, I'm going to skip that one. I'm going to go to this one. So this is an example of 2 genes that are related to breast cancer and ovarian cancer. So this is just like the blue squares or rectangles are representing exons from that gene. And these lines here and these letters represent point mutations that have been identified on those genes. So if we see all the exons here, there are many mutations that have been identified. And in the cases where you have a lot of small red points here, it's very frequent mutation. The ones where you have only 1 point, it's a less frequent mutation. But anyways, what I wanted to tell you is that so mutations can occur at any point in a DNA sequence, in a gene, but depending where it occurs, it will be the effect it has on the function of that gene. So if we have mutations in the coding region, what will be the result of a mutation in the coding region? And what will be the effect of a mutation in the regulatory region, the promoter region? So what do you guys think about what happens if a gene has a mutation or a series of mutations in the promoter region, what will happen or what could happen to that gene? Anyone has an idea?
The gene could become up or down regulated.
Right. Elisa, you wanted to say something?
That's what I was gonna say.
Right. So it can be either upregulated or downregulated. Okay. And what may happen if those mutations are in the coding region?
There's a change in protein function, possibly.
Okay, it could be a change in the protein function. So there are different types of mutations that can occur in the coding region. So if we look at this table here, so on the left-hand side, we have just a triplet of TTC. So that is going to be translated into AAG, and that's a codon for lysine. So if we have a point mutation that it's silent, that will change the sequence of the DNA at the RNA, but there is no change at the protein level. So that's a silent mutations. And there are a lot of those mutations going on in our cells all the time. So we don't realize, and nobody will see that we have that mutation because it's silent. Nothing happens to the protein, nothing happens to the function of our cells. But what if that mutation changes or results in a change in amino acid? It's what we have on the right-hand side, the missense mutations. Those can be either conservative or non-conservative. And what does it mean? So first, missense means that the amino acid is going to be changed. In contrast to the silent mutation, where there is a change of the DNA but no change of the amino acid, and the missense mutation, we do have a change at the protein level. So the amino acid, the lysine, will be changed by arginine or by threonine. And the conservative or non-conservative means that if you see here, this amino acid here, the arginine, it's a basic amino acid similar to the lysine. So the fact that it changes by another amino acid that has similar chemical properties to the original one, it means that it may not change a lot, at least the interactions, intramolecular interactions between amino acids in the protein. But if instead of arginine, you put threonine, which is a polar amino acid, that would change the interactions in intramolecular interactions of the protein. So that may result in a change in the 3D structure of the protein. And sometimes those tiny changes, but one amino acid change may really change a lot the function of the protein, because that can happen in positions that are crucial for stabilizing the protein. Or maybe if that change is in a pocket of the protein where there is interaction with the ligand, then the interaction may be lost, or the affinity can decrease or increase. So the answer to the question, what happens if the mutations occur in the coding region, is it depends on what kind of mutation it is. If it's a mutation that is silent, or it's a missense mutation, or a nonsense mutation. So what is a nonsense mutation? Basically, it's a mutation that will introduce a stop codon. And what is a stop codon? So when the DNA is transcribed into RNA, and then that RNA goes into the ribosomes for protein synthesis, then the protein synthesis will stop earlier than it should. So stop codon will mean that the protein will probably be truncated. So it's not going to be synthesized the full size. So that may, as you can imagine, it will have even more dramatic effect in the protein function. This is just an example I took from this. You have probably heard about the RAS oncogene. Do you know RAS oncogene? So do you know what oncogenes are? Right? So who can tell me what oncogenes are?
Cancer-causing genes, or cells that just continue to mutate and divide uncontrollably.
Okay. Anyone else? Any other idea of oncogenes?
I
It's
remember. Oh, sorry.
okay. It's regulate cancers.
Okay.
Okay,
Concept.
I remember that this is a kinase that is either hyperactivated in cancer, and this kinase is related with the activation with the MAP kinase in cancer that promote the cell proliferation, invasion, and migration.
Okay. So basically, when you talk about oncogenes, these are like genes that we already have in our cells, but when they are activated by mutations, they will promote the formation of tumors or the development of tumors. And in the RAS gene is one of those tumors that has been identified by carrying mutations in many different tumors, human tumors. And actually there's like one type of mutations, like the G12V mutation. It's a change of a single amino acid in the protein that will render that protein not hyperactive, but constitutively active. So these proteins, the RAS proteins or the RAS family proteins, are signaling molecules that are activated by, they are known as switches, molecular switches, because there are external signals coming through a receptor in the membrane, and inside the cell, these proteins take that signal and transduce the signal to some other molecules, and then they go back to the inactive state and they will activate it again. So there go rounds of activation, inactivation, and then if one of these mutations occurs, that protein will stay active all the time. And let's imagine that the signal that RAS is sending to the cell is divide, divide, divide. What happens then if there is a mutation and that signal is all the time on? So the cells are going to start dividing very quickly and nonstop. So that's a very, very it's one example of what can happen if there's a point mutation in a gene like the RAS gene. And this is just an example of the signaling cascade where we have these receptors at the membrane. They will activate RAS. RAS will change GDP by GTP. So this is the active form of RAS. And that active RAS will signal downstream to activate other pathways that will give rise to proliferation. And what happens if we have the G12V mutation, that protein is constitutively active, and then we have this MAP kinase pathway active all the time, and then it may induce a lot of changes in the cell behavior that may give rise to tumors. So there's also another important concept that it's there are some so-called hotspots for mutations, which are regions in the genome that have been identified to carry mutations very often. It means some mutations may occur anywhere at any point of the genome, but there are some hotspots where in many cells, many organisms, or many humans or tumors or whatever, if you look at that particular position, there are mutations in that particular region. And this is just an example of the melanoma-related mutations. I'm not going to go into details, but if you look at these scales here, the red bars, so when these guys looked at the genome-wide mutations, so they look at the whole genome and they compare how frequent were the mutations in different sequences on the genome, they found that these particular region here, those mutations that are highlighted in gray color, they were very frequent. When looking at these frequencies here are when looking at the frequencies compared to the whole genome. But what happens if they look at genes that have very high expression, very highly expressed genes? So they found that the probability of mutations on these particular nucleotides is very, very high. So they ended up saying, okay, it seems that this is like a hotspot for mutations, particularly in genes that are found in a very highly expressed in melanoma. So then what they did to see what further going to explore this, they exposed cells to UV light, and then they looked how this UV light may induce mutations in the genome, and they look at some particular genes that are in this category here. So they found genes that were highly expressed and that had mutations very often in those particular regions. So they took cells, they exposed them to UV light, and then they looked for mutations on those particular regions. And as you can see here, the top panel here, we have the control cells, and down we have the cells that were exposed to UV for 5 weeks, after 5 weeks or after 10 weeks. And as you can see, that there is an increase in the number of cells that carry those mutations in this particular sequence. So that demonstrates that if you expose the cells to UV, instead of having DNA mutations all along the genome, there may be some regions that are more prone or more susceptible to